right, ladies and gentlemen, welcome to our last couple of screencasts on cell division. We're going to start talking about meiosis. This is where we left off. Before we get too far into meiosis, let's just label each stage of meiosis 1 and 2, and I promise it's going to be easy. It's going to seem familiar. This first one is interphase, right? Because interphase happens before meiosis and mitosis. It doesn't matter. Interphase is always that 90% of the cell cycle where G1S and G2 occur. Then you have prophase 1, because this is meiosis 1 up top. Metaphase 1, anaphase 1. And again, notice all the same characteristics are here. In prophase, the nucleus is breaking down, our centrioles are moving apart, forming the spindle. Our chromosomes are lined up across the middle. The only difference here is that we have homologous pairs lining up. Homologous pairs are lining up. And I encourage you to go back and look at the definition of homologous pairs. And then in anaphase, homologous pairs are pulled apart. Notice each chromosome at this point is still comprised of 2 chromatids. That's a big deal. Then we have telophase 1 and cytokinesis, obviously, because we go into 2 new daughter cells. Then in meiosis 2, a big difference here between meiosis and mitosis is in meiosis 2, we have 2 cells going through this process of cell division. We have prophase 2, metaphase 2, anaphase 2, and lastly telophase 2, followed by cytokinesis once again. So let's talk about each of these phases individually, because there are some differences between these phases and the phases of mitosis, in particular when it comes to chromosome count and number of cells. Alright, so let's look at meiosis 1. In interphase, chromosomes replicate. We know this, right? S phase, DNA is duplicated, same thing. And the centromere holds the 2 sister chromatids together. In prophase 1, chromosomes are going to shorten and thicken. They're going to become more chromosome-like, right? We can see they look much more like the chromosomes we're used to seeing here. And each chromosome is actually going to pair up with its corresponding homologue, right? Just like on that karyotype picture I showed you a few slides back. The number 1 chromosomes pair up, the number 2s, the number 4s, number 20s, number 10s. They all pair up together, and that's what these structures here are, right? This is one duplicated chromosome, that's another duplicated chromosome, and they are homologues to each other, meaning this red one could have came from dad and the blue one could have come from mom. But when you have these homologous chromosomes paired up, we call it a tetrad. We call it a tetrad. The prefix tetra stands for 4, and there are 4 chromatid arms here, right? 1, 2, 3, and 4. So that's why we call it a tetrad. And then in metaphase 1, tetrads line up across the middle, which is exactly what we see here. And in anaphase 1, the tetrads are actually broken apart. Homologous pairs are separated. In meiosis 2, now keep in mind, the spindle complex still attached to the centromere of those homologous pairs. The spindle complex shortening is what pulled them apart.
Main office. Mr. Journey, please come to the main office or call the main office.
And all of the same things are happening in that meiosis 1 phase as mitosis. It's just happening to homologous pairs of chromosomes rather than individual duplicated chromosomes. All right, so let's look at meiosis 2. So in telophase 1, sorry, got to finish up meiosis 1, the cell is going to separate into 2 cells. Okay. Meiosis 1 does result in 2 haploid or 1N daughter cells. Okay. However, remember, haploid only means 1 set. Okay. Haploid means 1 set. At this point, notice each chromosome here is still comprised of 2 sister chromatids. So we haven't reduced chromosome number, right, between meiosis 1 and the start of meiosis 2. And the point of meiosis is to reduce chromosome number. Now, we went from 92, right, chromosomes, or I'm sorry, 46 chromosomes, 92 chromatids, to now we have 46 chromatids in each cell, okay? But remember, we need to get down to 23 chromatids per cell. And that's what this meiosis II is going to do for us. So let's follow this out. In prophase 2, the pairs of sister chromatids, all right, now the duplicated chromosomes, all right, they're going to start moving toward the center of the cell. The nucleus started to reform, but then it gets broken down again. The spindle complex is beginning to form between the centrioles. Metaphase 2, just like in mitosis, pairs of sister chromatids line up at the center of the cell. All right, this looks much more familiar to us than previous stages, like in meiosis I. Now you can see here, when these spindles shorten, it's going to be the chromosome itself, the duplicated chromosome, that gets pulled apart at the centromere. And this is where we are going to reduce chromosome number to 23 for the reproductive cell. Anaphase 2, the pairs of sister chromatids are pulled apart at the centromere, and they move to opposite sides of the cell. And in telophase 2 in cytokinesis, we're going to have a result of 4 new cells that are all haploid, and they are all going to be genetically different. All right, if these were 4 egg cells, they'd all be genetically different. If these were 4 sperm cells, they'd all be genetically different. And I'm going to explain to you how they would be genetically different. And I don't know if you've noticed, but notice the colors of these chromosomes. All red with a blue arm, all blue with some red arms. Something is happening with the genetic material between these chromosomes, and that's what allows for an intense amount of genetic diversity in this process. All right, so what is the importance of meiosis? Well, we're going to start the next screencast with that, folks. See you in the next one.